So this last video, I would say it's the interesting part of this chapter that you finally get to see how cell cycles are controlled and how it has an effect, if uncontrolled, to cause disease. Okay, so usually cells go through cell cycles to do what? We talked about this, mainly to go from one to two, right? And actually, when cell is going through the cell cycle, it has points during the stages, okay? And the main thing is it should only go forward and it has to go through every stages. It can't just, oh, I like G1, I like S, and I'm going back to G1 and then S, because the cell can't keep copying its DNA without dividing one into two. So cell is highly controlled, and there's two irreversible points. One is once you replicated your DNA, you have to move on and actually split. Okay, so you have to split your sister chromatids. And let's also talk about checkpoints right here. Checkpoints are specifically certain points within the cell cycle that the cell checks for things, okay? For example, cells check during G1 into S phase. So these are the three checkpoints. When the cells are going G1 into S, it first checks whether or not its DNA is good. Is it damaged? Is it mutated? If it is, then it shouldn't go into S. It shouldn't one become two, because the genetic material is damaged, and for it to copy another copy of itself is actually forming another mutant in the body. So that's not good. Okay, so that's the first checkpoint, G1 into S. What does it check? It checks for DNA damage and mutation. If there's damage or mutation, the cells should check, stop, and actually fix it. That's G1S. The second is G2M. So remember, G1 into S. Okay, my DNA is good. I'm going to copy in an S. Then it's G2, right? Now, G2M checkpoint is when the cells have successfully made the replication, but before it goes into split, it's actually going to check if it copied right. That means during S phase, I see AT, I put AT, right? But then what if I didn't copy correctly? Okay, so remember, DNA replication is semi-conserved. If you see A, the other strand should be T, but what if somehow the cell decided to put G and it's wrong? Then you fix it right now in G2M. That's another checkpoint, very important. Again, we want to be faithful to exactly what the cell sequence or the genetic code should be, the DNA sequence should be. Okay, so all these are very important checkpoints. Last checkpoint is during metaphase. What happens during metaphase? Remember, we just drew that out. All the sister chromatids line up in the middle, and you want spindle fiber from each side holding on to each sister chromatids. And then when you split the sister chromatids, correct? So the spindle fiber from the right should hold on to one sister chromatid. The spindle fiber from the left should hold on to the other spindle fiber. And when they split, then I get one of each. Well, I have to check for that. What if that didn't happen? I'm loose, I didn't attach. Then anaphase comes, and when I pull, I'm gonna pull two. That will cause genetic imbalance, okay? So late metaphase, you also need to check. Check what? Ensure that all chromosomes, each chromosomes, have spindle attached. So when I split, this comes here, this comes here. Not that this one didn't have any attached, and it's just gonna go with that one because they are stuck together. Okay, so what are the three checkpoints? One is G1 to S, G1S. What are we check for again? Remember, G1 is growing. Before I replicate, first of all, you check for DNA damage, is it mutated? Then you copy. G1S into M. What are you checking? Did I copy correctly? That's G2M. And then this spindle check, what's the M phase checking point? Metaphase, I'm checking that I have spindle from opposite pole on each sister chromatids. Okay, so that's very important. All three are important because what if it doesn't work? What happens when it doesn't work? That's when cancer happens, okay? That's when the cells aren't checking itself and it just keep growing and keep growing. What's worse is it keep accumulating mutations. G1S, it didn't check if it has mutation. It just copied another version of itself. G2M, it should stop because it copied wrong and it continued to divide and make another copy of itself. Metaphase, right? It's supposed to have one of each, but it didn't. It has two and it's still divided. So all these could cause problem, especially cancer. So cancer is a failure of cell cycle control when cells had a mutation, a DNA damage, and it didn't stop. Ideally, a cell should stop. And in fact, your cell is designed that if it stops, it has a mutation, it should stop from replicating itself. That's one thing. It should fix itself. And if it can't do that, it should kill itself, called apoptosis, cell suicide, okay? So these are actually embedded into the cells for your benefit. But if it doesn't work, then you got cell proliferation out of control with bad DNA, okay? And there are two separate types of DNA that actually controls these cell cycles, okay? One type is called tumor suppressor genes. The other type is called proto-oncogenes. Okay, so I'm gonna give you an analogy. Pretend we're going through cell cycle. Pretend going through is like driving. You keep going forward. G1S, G2M, G1S, G2M. You just keep going through your cycles. Yeah, and cytokinesis, right? G1S, G2M, and then M contains mitosis and cytokinesis. You're just going through all these cycles. Now, usually, if you have a mutation, then your tumor suppressor gene working properly should say, hold on, stop, you have a mutation. That's what tumor suppressor gene do. Everybody has it. If it functions normally, it's going to put on a brake. The analogy is brake. It's going to put on a brake and say, hold on, stop, so you don't get into a car accident, okay? Now, proto-oncogene, on the other hand, is saying, go forward, keep going forward. That's the gas pedal, okay? So normally in your body, there are these two types of genes. One type that says, hold on, let's check for it. One type that says, no, you're good, go ahead and multiply. You have both, okay? When everything works well, it's great because you should be dividing and that gives you renewed cell or you give you growth. You should be stopping when there is mutation. So when everything goes well, tumor suppressor gene and proto-oncogene should be there. Now let's review. What are the two major groups of genes? Can you name them? Tumor suppressor, proto-oncogene. Now, example of tumor suppressor gene is a protein called P53, okay? And what it checks for is during G1 phase, so it's a G1S checkpoint protein, if there is damaged DNA, P53 is going to say, stop, don't go forward, fix. Well, what if the cells can't fix the mutation? Then P53 says, please kill yourself. And that's actually for your own good, so you won't become a tumor. Now, if P53 is not properly working, then you have a problem. Then most likely you can have cancer because your cell is not supposed to divide, but it didn't have to break, you see. So it rams forward and it gets into a car accident, okay? So to prevent the development of a mutated cells, you need a healthy P53. But if you have a P53 that's absent or mutated and cannot work, it's like having a brake pedal that doesn't function, okay? Or it's missing. I don't have a brake, or it doesn't put on the brakes, okay? So that's an analogy. So let's look at how this works. Normal P53, DNA damage. So this is double helix DNA. It has a damage. P53 comes in, checks for it and say, you got to repair. It repairs. Perfect. Then you can go on because now you're healthy, you repaired 1 into 2, 2, and keep going. If you cannot repair, then the cell division stops and P53 triggers you to then disrupt yourself, which is good because you cannot fix it. Now let's look at cancerous situation when you have an abnormal P53. Again, this abnormal situation is what we call loss of function. That means the brake pedal does not work. That's when you get into a car accident. But it does not work is not because it's stuck and keep braking. Does that make sense? Because if you keep brake on your car, you don't get into a car accident, you just stop there. A loss of function means the brake no longer works and you keep going forward because you cannot stop now, and then you drive through. So let's look at the situation here now. You got a mutation, you're supposed to stop, but you're not stopping. You say, what the heck? Let's go forward. My P53 is not working. So you split, and what happened? You have an abnormal cell. This is a mutated cell, and the cell no longer stops. It just keep dividing with the mutation. What does that mean? That means you keep gaining more and more mutation. As you copy, as you do stuff, the cell doesn't ever stop. It just keep kind of accumulate more and more mutation, and eventually it develops into an uncontrollable growth cancer cell. Okay, proto-oncogene. What did I say? The analogy is tumor suppressor is like the brake. If you lost a function, the brake doesn't work, then your car drives through. So if P53 does not check, then the mutation continues. That's one type called tumor suppressor. The other type is proto-oncogene. The analogy is gas pedal. This type of gene is not about stopping. This type of gene is about going forward. It's a grow, grow, grow. Okay? So normally, you do have proto-oncogene. What are these? These are like those growth receptors on the cell surface. When growth factors, NGF or EGF, okay? When you have those growth factors coming in, okay? Remember they're ligands, and your receptor binds to them, then it relays. Remember chapter 9? Say grow, grow, grow. That's the proto-oncogene. It's good. It's there, so you will grow, you will divide. But what if it mutates? What if it mutates and now, without the ligand, it keeps signaling growth? That's like a stuck gas pedal. That's a gain of function. So that's different between proto-oncogene and tumor suppressor gene. When proto-oncogene gains function and keeps signaling, even without, so nobody is telling it to grow, but somehow it's stuck in the receptor activation situation. It keeps signaling, signal transduction, keep growing, then the cell just keep dividing, even without the body sending out growth factors saying grow. Okay, so then it turns from proto-oncogene to oncogene, and it actually signals growth. So some encodes receptor for growth factors, and then it signals on, keep growing. Okay, some encodes signal transduction protein down below. Signal transduction, even without the growth receptor saying growth, there's no growth factor. It just keeps signaling, and the cell didn't know. It's like, oh, you're telling me to grow, so I will keep growing. Okay. The thing is, you only need one copy of proto-oncogene, which means you always have 2 copies of every gene, one from dad, one from mom, right? If we are talking about P53, tumor suppressor, this brake doesn't work. Hey, I still got the P53 from mom. It works. So I am still going to stop when I need to stop. Okay. But proto-oncogene is a gain of function. I said, keep going. It doesn't matter that the other one works and the other one says, no, I'm not going to say keep going. But this signal one keeps saying keep going, then the cell will keep going. Okay. So you only need one to cause uncontrollable division. Okay, so that's it. Okay, so from this video, you mainly need to know cell cycle controls cell division, and it's highly controlled. mainly with 3 checkpoints: G1S, G2M, and then the M checkpoint. And what do they check for? Then you got 2 groups of different genes that actually controls the cell cycle. When they're working properly, it's great. Proto-oncogene says grow when you should grow. Tumor suppressors gene says stop and fix when you should. However, when these 2 signals somehow doesn't work, then you have something called cancer. But they're not working is kind of different. Tumor suppressor is a loss of function. Imagine braking the brakes of a car. If it can't stop the cell from going forward, then you have a problem. Proto-oncogene on the other side is like the gas pedal of the car, okay? When it has a gain of function, then you have a problem. When it says go, 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 okay? When it has a loss of function and it just doesn't function anymore, that's fine. It just doesn't tell the cells to grow, then you don't have cancer, okay? So kind of think through these 2 kinds of cell controls. And if it's not clear, then make sure that you go back and kind of think through what is gain of function, what's loss of function, what's tumor suppressor, what's proto-oncogene. Okay, this needs to be super clear for you to fully understand the control of cell cycle. Okay, that's it. This concludes chapter 10.